All right. Who wants to take this one? Genetic variation in a bacterial population cannot result from...
Meiosis.
Yeah. Okay, which one of these cells, A or B, is considered F positive?
Be cell A.
Why cell A?
Because it's the one infecting the other cell with the F factor, whatever it's called.
Yeah. Okay, so it's the one donating. Yeah. So cell A. Which of the following bacterial roles typically does not involve symbiosis?
A pathogen.
Decomposer.
I'm going decomposer. Why not pathogen?
Well, a pathogen's role involves a kind of symbiosis, doesn't it?
It does. Symbiosis, symbio just means living together. Doesn't mean happy, lovely, mutualistic. So pathogens, they're actually symbiotic. So decomposer for that one. Photoautotrophs use
Light and CO2.
Well, in what way?
Light as energy, CO2 is carbon.
Yeah, light is energy, CO2 is carbon. All right, not true.
Only bacteria has histones.
Okay. How'd we do? We did good. 10 out of 10. Is there anything else about the quiz, or does that cover it for you?
I came in while you were on question 2. Could you go over question 1 real quick?
Bacteria don't do meiosis.
Okay. Thank you.
Yep. All right.
So was decomposer the answer because the system's already dead, so you have an external organism acting on an already dead system?
Yeah, it's not two things living together.
Okay.
Yeah.
Thank you.
Yeah. So if you've got a tapeworm, that's really bad for you, but it's still a symbiosis because you're living together with your tapeworm. Okay.
Sorry, I think this is just a personal misunderstanding of symbiosis versus, oh my gosh, not parasitism, but not symbiosis either. Does symbiosis have to be mutually beneficial, or can there be kind of that middle ground where it's like, it's not killing you, but...
You know, it can even be killing you and be a symbiosis.
Okay.
It exists on a spectrum all the way from parasitism to mutualistic.
Is there a different word for like mutually beneficial versus like one's harming, one's hurting to varying degrees?
Yeah, so mutually beneficial is just called mutualistic symbiosis.
Okay.
And one's getting a benefit, but the other one's getting nothing, not harm or benefit. Then it's called a commensal. So commensalism.
Commensalism.
And then if one is being bad to the other one, then it's parasitism. And so it kind of exists on that gradient. I think in like everyday speak, in English, we use the word symbiotic to kind of mean like mutually beneficial.
Mm-hmm.
And I think that's where the confusion kind of comes in.
Totally.
Yeah.
Thank you.
No problem. Okay, so it turns out last class period on Friday, you're asking about the test, like time limits and when it's available and all that. I was misunderstanding the question there. There is a time limit on the exam once you start it. I was thinking you were saying like how long is it open? So it is 68 minutes once you start the exam, but you're free to start it anytime during that day. Does that make sense? I apologize for that. Could misunderstood.
How many questions are there on it?
34. So you get 2 minutes per question. The bulk, maybe about half the questions are about like quiz questions, right? Just like simple recall, multiple choice, right? Do bacteria do meiosis, that kind of thing. And then there's maybe 8 or 10 questions that are kind of a higher level. So 68 minutes should be plenty of time. That's more time than I gave last time I taught. Last time it was just the 50 minutes of class to take the exam, and all but one person finished well before that.
With the
Are we
quizzes,
going to be asked about quiz?
I'm sorry, what? I think two of you were speaking at once.
About quiz?
Will you be asked about
quiz?
Let me turn up my volume. Cladistics? You're kind of cutting out a bit. Okay. Yeah, I mean, cladistics is just phylogenetics. That's kind of the main thrust of this first exam is, mechanisms of evolution, how to read a phylogeny. A cladogram is just a certain type of phylogeny.
with the quizzes, it has that kind of setup where we have to, where we can't go back once we answer a question. On the test, is it going to be like that, or will we be able to go back to other questions we've already done?
You should be able to go back and look through it again. It's kind of just like all open. It's one big. So you get all 34 questions at once in one giant sheet, essentially.
Perfect.
You can scroll.
Okay, thank you.
Yeah. All right, any other questions about logistics or whatever, this being our first exam?
Taken online, right?
Yeah.
Just wanted to make sure. Cool.
Canvas. Yeah, it'll be on Canvas. Yeah. No lockdown browser. None of that.
Thank you.
No creepy dude in India asking to show your ID or tracking your eye movements. It's all fine.
Is my FBI agent allowed to help?
My rule is that you can't get help with anybody else. You can use the internet, you can use your notes, your textbook.
Okay, I'll tell my FBI agent no.
Yeah, tell him no. I realized that some of you may break that rule. I can't stop you. I don't have enough mental bandwidth to even care, honestly. Like, if you're that desperate to cheat and you want to get together with somebody and take the exam together, fine. Okay. Just don't tell me about it because it's against the rule. Whatever. We all have plenty to deal with right now. We don't need to be fretting about this too much. Okay. Well, shall we talk about prokaryotes?
Sure.
Ready? All right, well, you've got your main topics on the website. Pretty basic look at them, which is disappointing because like probably 85% of all biochemical diversity on Earth is tied up in prokaryotes, and we have allotted to us essentially 2 class periods to go over them. It's pretty lame, but that's okay because we've got a microbiology class where you get to learn a bunch more about them. So this is pretty surface level stuff. You're not going to be memorizing phylogenies of the different prokaryotic groups or whatever, any of that. Just taking a look at them, how they're alike, how they're different from eukaryotes, which is what everyone always thinks of when they think about biology, is plants and animals. Okay. So to start out with, the word prokaryote is not biologically sound. Okay. They come in these three shapes here. We've got the Skittle, the hot dog, and the spaghetti shape. They tend to be single-celled. Not always. They do break all the rules possible. There are multicellular ones. You've seen one in lab. What was that? Anyone recall? You met a prokaryote.
You mean like the cyanobacteria?
Cyanobacteria, right? And so that, I think it was Nostoc that you looked at. That's actually multicellular, and it's got cell differentiation with the heterocysts and all that. And so prokaryotes tend to come in these three shapes. There's also some that are star-shaped, which are ridiculous, okay? But the textbook definition of prokaryotic cell shape gives you Skittles, hot dogs, and spaghetti, and that's basically it. That is 99.9% of them all, okay? And they do tend to be single-celled. They're a much simpler form of life, and prokaryotes make up two domains, which is why the word prokaryote is biologically useless. What is it about that term that we don't like here in phylogenetics? The clue is here on the slide.
Is it because archaea and eukarya are sister and not eukarya?
They are sister, and so what's that make the term prokaryote, or the group prokaryote?
Would that be a common ancestor, or no?
Okay, so bacteria and archaea are what we call the quote-unquote prokaryotes. They're not sister to each other. And so if we want to draw a circle around them and make a group...
Paraphyletic, right?
It's paraphyletic, right? So we're looking at this common ancestor and then leaving out eukaryotes. So the common ancestor of archaea and bacteria is the same as the common ancestor of eukarya and bacteria. And so prokaryote, the term, is actually paraphyletic. Okay. This is fairly new knowledge. Happened in my lifetime, I remember this. But there's really no reason to use the term prokaryote anymore. And once you get into microbiology, I'm sure it's not allowed. Okay, because archaea and bacteria they look alike maybe, but they do a bunch of different stuff. Okay. For instance,
isn't the basic definition of... Oh, sorry.
No, go for it.
Isn't the basic definition of prokaryote it was defining without a nucleus, right? That's why they grouped them together.
Yeah, prokaryote means prior to nucleus.
Okay.
Yeah, and so they have that in common. That's nice. And that's why when we saw them microscopically, you know, we knew there were things different about them. When we use an electron microscope, like the cell walls are different. But they're single-celled, really simple. They don't have a nucleus, and so we classified them as sister to each other until phylogenetics came along. And once phylogenetics came along and we made this tree saying that archaea and eukarya are sister, they actually started to make more sense. We'll see that in a second. Okay. So archaea don't have peptidoglycan in their cell wall. Bacteria do. And then in their membranes, bacteria have this ester linkage right here, that extra oxygen. And archaea have an ether linkage. All right, based on our tree here, our phylogenetic hypothesis, what would you predict about eukaryotic cell membranes?
That they have no peptidoglycan.
No peptidoglycan, but that's their wall. What about their membrane?
They have the ether linkage thing.
Yeah, and so when we do look at eukaryotes, we see that we, humans, also have this ether linkage, the same as archaea. Okay, so there's things in common between eukaryotes and archaea that are made sense of when we have this tree, that before were quite baffling. Okay. We've got a bunch of metabolic pathways. These are the ones that I know of or could found in archaea. What's missing from these?
There's no photoorgano.
No photoorgano. And we're missing another photolitho. What's cyanobacteria? What's its metabolic pathway?
Isn't it a photo autotroph?
Yeah.
No. Yes?
Yeah.
Okay.
Photolitho auto. It's the same as a plant. Okay. And we'll see why in just a second. But photolitho auto is what plants do. It's what the green stuff does. We don't find that in archaea. But we do have photolitho hetero, which is a really weird one. And so they're using sunlight for energy. They're using water for electrons, but their carbon source isn't CO2. They're actually eating carbon. Utah is actually famous for these. So this is Great Salt Lake. And what was it, like 50, 60 years ago when they built the railroad bridge across the lake? It divided the lake into two halves. And this is the color of them seen from space nowadays. What's different?
The compound that they use to absorb light.
Yeah.
Yes? Okay.
Yeah. So they don't use green pigment to absorb light. They use rhodopsins, these like pink pigments. It's the same thing used in your eyes to detect light. Okay, so same, just like eukaryotes, they use these rhodopsins, and that's what's making it pink. And so the northern half of Great Salt Lake is ultra salty, like way saltier than the southern half, and these things are just thriving up there. They're called halobacteria, which is unfortunate because they're not bacteria, they are archaea. Okay? They were named before we knew that. So these are archaea living up here doing their photolitho heterotrophy, and that pink pigment is them absorbing sunlight for energy.
Is
yeah.
The way they absorb sunlight more or less efficient than chlorophyll?
I don't have an answer for that. I don't know. I can tell you that it's efficient enough for them to get by.
Yeah.
Maybe that doesn't.
Does it use the same process converting CO2 and water and all that stuff and turn it into glucose?
Not
Does
CO2.
It use the same process?
No, it's not CO2, and it's not the same sort of photosynthetic pathway. They don't have rubisco fixing carbon from the atmosphere because they're heterotrophs. They're actually getting their carbon from organic sources. They're just getting energy from sunlight. So there are a lot of pigments that can temporarily absorb sunlight and makes them vibrate and move around and shift electrons. That's what photosynthesis in plants is doing. It's just chlorophyll is that pigment. Here it's rhodopsin.
I had a question about that actually.
Yeah.
You said that was caused by they built a railroad, you said?
Yeah.
Okay.
Like, and there's very little flow between the north and the south. And so Jordan River actually pours in here, and so they've got more freshwater input in the southern half, and there's only a few little spots where it kind of mixes, and so the northern half has just become way, way saltier over the last 50, 60 years.
We should take a field trip out there as a class.
I like it. The northern half, I think, is cooler than the southern one. The southern one, I don't know if you've been there, it's full of brine shrimp and it stinks.
Yeah.
Real bad. It's disgusting. You gotta wade through like
the-
piles of rotting brine shrimp. The
It's the
northern
Army
half,
Corps of Engineers that built that, and they tried to pump water and like keep the cycle in the lake going, and then they just gave up.
yeah,
So
yeah. There's still like the old pump station here at the end. From what I understand, they spent a number of millions of dollars trying to do that, and then just kind of quit.
yeah, it's because the tracks because of the nature of that soil, that the tracks keep sinking and they have to build them up, and so it kept breaking the pipes, and so they just said, "What the heck? Who cares?"
Yeah, well, it's created an awesome laboratory for us. So there's a lot of actually pretty interesting research because we've got this salinity gradient. That's one of my projects that I've kind of given up on over the past year and a half because, something about a pandemic or whatever. But hopefully I'll get back to it. This northern half is ultra salty and not much can live there. It's too salty for brine shrimp. And so like the main organism that's living there is this halobacteria, this salt-loving halo archaea. All right, so there's these differences between archaea and bacteria, but there's also some stuff in common. And this is why we started, classifying them originally as bacteria. We didn't have that third domain. Okay, we've got a circular chromosome. This is way different from the eukaryotes. We got really fast generation time and really low mutation rates. By low mutation rate, I mean that when they're copying this circular chromosome, their DNA polymerase adjacent proteins that do the proofreading are really, really good. Okay, so they have an order of magnitude fewer mutations than an animal or a plant, for instance. So, question for you. How did they evolve so quickly if mutations are so incredibly rare?
Large populations and low lifespan, or small lifespan.
I mean, there can be a lot of generations in a small amount of time.
Yeah. Okay. Yeah, I think that's probably what you were trying to say is really fast generation rate. So yeah, short lifespan, kind of translate that into they reproduce very rapidly. And so they can divide, some bacteria every 20 minutes. And so you can see this exponential growth curve where you start with like 10 cells and you wind up in a couple hours with 650. Okay. Here's E. coli, that's got a 30-minute generation time. That's really fast. Like incredibly fast. How long does it take a human? For a human to double, what's the generation time?
Like 20 to 30 years.
30.
Yeah, I mean, you got to have 2 kids to double yourself, and so you're probably talking 20 at the kind of the low end. That's really, really long time compared to these bacteria and archaea. And so we're talking about thousands and thousands of generations. In thousands of generations of humans, it'd be a lot longer time span than 11 days, but we could see the same amount of evolution in that time. Okay. And so they do mutate very rarely, but they evolve very quickly because they're constantly copying that DNA, right, that circular chromosome. And another reason is because they do this lateral gene transfer. Sometimes you'll see this called horizontal gene transfer, same thing. All right, so it's a synonym, lateral or horizontal gene transfer. All right, what is that?
When two different organisms exchange DNA or combine DNA.
Okay. Two different organisms exchanging DNA. Yeah, that works for me. It's different from vertical gene transfer. How?
Isn't vertical gene transfer as in your descendants?
Yeah, your descendants. And so if a bacterial cell divides through binary fission, it copies its chromosome, divides. The new daughter cell, that's vertical, right? It got its DNA that it has from its parent. So that's kind of top down, this vertical transmission. Lateral is you're sending or receiving genetic material from things that aren't directly related to you. Okay. And it comes in three flavors. We've got transformation, transduction, and conjugation. Okay. Transformation is just when you're picking up DNA from the environment that's kind of floating around. Okay. So you're a bacterial cell floating around in a soup of broth or whatever, and there's some DNA that got excreted by another bacterial cell, and you take it up into your cell and incorporate it into your chromosome or into a plasmid. Right? Give me some pros and cons for that.
Sorry, the bacteriophage picks it up?
No, it's just like raw DNA floating in the environment, in the soil or in the soup that you left in your fridge for too long. There's DNA floating there, and a bacterial cell can actually just take that in.
I guess.
So
I mean,
actually,
it's kind of random. So, go ahead. Sorry.
So you're saying random?
Well, yeah. I mean, assuming that they just randomly pick up whatever naked DNA is around, then it could be either a beneficial or a negative mutation, as it were, that occurs.
Yeah. So it can be bad, right? So that could be something nice. Like it could be a bit of DNA that gives you resistance to an antibiotic that's present, right? That'd be great. But it could also be a bit of DNA that's bad for you, that makes you more susceptible to antibiotics. Okay, so how do you distinguish between the two?
I imagine you could look at the proteins it's holding on to distinguish against the different things, but
okay, well, it's not got any proteins with it necessarily. It's just DNA. And I mean, DNA is DNA. It works the same way in every organism. We have the universal genetic code. And so, I mean, this is how we make insulin now, right? We used to take it from rabbits and pigs and stuff, but now we just took the human insulin code and put it into E. coli. And so now we have industrial vats of E. coli making human insulin for us. Because DNA is DNA. It's just like computer software. It's ones and zeros. It will run on whatever system you put it in.
assuming that, then you could essentially code it to be non-resistant to certain antibiotics.
Yeah.
So depending on what you're asking, couldn't you distinguish against what bacteria have it and what don't by just placing it inside a solution of that antibiotic?
Oh, yeah. So you, the human, could distinguish that, but what about the bacteria?
Oh.
Right. So, I mean, it's floating around. There's a piece of DNA, and it just takes it up, just like an idiot. I mean, it could be bad, right? And so how does
Bacteria
the
be hungry.
Yeah, right. They're just sucking up this DNA. What prevents them basically from just taking up the bad DNA and passing that to everybody else in the population?
Would it be the survival of the fittest idea, where if they pick up a bad DNA, it's going to make them unfit for their environment?
Yeah, so this is the thing, is that bacteria can't always distinguish whether it's going to be good or bad. There are cute little ways that they can, but in general, if it's random DNA and you're taking it up and it turns out to be bad for you, what happens? Well, you die. And so you're not passing it along to your offspring. So it kind of is a problem that solves itself. Okay. If you take up the good DNA, you're better off and you pass that along to your offspring. If you take up bad DNA, you die, so you don't. And so there's selective pressure there. Okay. Now, transduction is lateral gene transfer, just mediated through viruses. And so pictured here is a phage. So bacteriophages are viruses that prey on bacteria. And so this phage lands there and it excretes some DNA into the cell. And it's basically the same thing. It's just mediated through phages. So that's how it's different from regular old transformation. So transformation, picking it up from the environment. Transduction, it's from the environment, but you're getting it through a phage. And then conjugation, this third way, is that you're getting it directed to you through this kind of sex tube, essentially. It's this pilus. Okay? So it's a tube from one bacterial cell next to you that grows in, conjugates with you, and then it sends DNA to you. But all three of these different vectors, whether you're transforming from the environment, transduction from a phage, or conjugation from another bacterial cell, they still run into those same problems of the DNA can be good or bad. All right. So have you heard of CRISPR-Cas9? Anyone?
I have heard of it. It sounds very familiar. I can't quite remember what it is.
Well, it just won the Nobel Prize, which is awesome.
Oh, sorry.
Yeah.
Is that what they use to like alter genetics?
It is. And it's not like we invented it. We figured out something that bacteria are doing, and we stole it from them, which is brilliant. Okay. And so, yeah, it's very deserving of a Nobel Prize, because now we're using this bacterial technology that we can edit cells in vitro. We can change genetic code. And so bacteria actually do have something kind of like an immune system, and that's what the CRISPR-Cas9 is. And kind of like our antibodies that can recognize a protein, okay, they've got little strands of DNA floating around in them attached to these CRISPR proteins that fit like a lock and key on certain stretches of environmental DNA. And so if a phage dumps some DNA into your cell, right, it splits up, it's single-stranded, and it's getting ready to incorporate into your chromosome, you've got something that's its reverse complement, so it matches right up to it. Okay, so you've got an A where its T is, and so forth. You stick to that, and then it just cuts it. Okay, and it's this 21 nucleotide key, essentially. And so bacterial cells can, through evolution over time, start to recognize some of the common bad stuff that's out there, so like viruses. Okay, so there is a mechanism, this CRISPR mechanism, that they can use to chop up bad DNA. But when we look at the sorts of stuff that are being passed around horizontally, laterally between prokaryotes, bacteria and archaea, the book talks about these two big ones in particular, because these are the ones that spread rapidly through any population of bacteria. Okay. And those are the R and F factors. An R factor is just some DNA code that gives you resistance to something. Okay. So it was named for antibiotic resistance, so we call it the R factor. Right. But it could be resistance to high salt levels or temperature or freezing or whatever it is. It gives you an advantage. And so it's a bit of DNA that confers some resistant advantage to you. So you're more likely to survive an environmental stress. Okay, that's an R factor. And then the F factor is the fertility factor. And that's the instructions to make this pilus, this little tube. Okay. What's the point of the F factor?
Gives them a better chance of passing on the beneficial R factor.
Definitely, much better chance. Okay, so if you want to help out your fellow bacterial cell, you just got this resistant factor and you're passing it on. You could just throw it into the environment. You could excrete a plasmid, these little circular short chains of DNA. You could send that into the environment and just hope, not that bacteria have hopes and dreams, but you could just essentially count on the bacteria around you picking that up before it gets broken down by DNases and stuff in the environment. That's nice, but maybe competing bacteria could pick it up also. A different species living there that's competing for the same resources. And so that's not really good for your population. So this fertility factor lets you directly transfer material. So the question here I want you to think about, why is this fertility factor so widespread, given what we've seen?
If you aren't fertile, you can't reproduce, so it makes sense that it would just get higher and higher.
You can reproduce. You can still do your binary fission thing.
Oh, yeah.
And this isn't true sex or meiosis or any of that. You're just sending a little plasmid, right? This may be 5,000 base pairs long, just this little circular chunk of DNA that looks like a tiny chromosome. You're just sending
Genetic
that.
variation and communication.
Can you elaborate?
This is mostly a guess, first off, but genetic variation, if I were to think of a way to like a plant can, or even we've got bunnies and they can like store genetic material for later use, and so I could see that being a potential use where like if there is something in their environment that they need to make a protein for, they have a genetic code to make that protein. Or since we're talking about bacteria and smaller things, potentially communication, saying like, hey, this is the state that I'm in right now. But I am even less sure about that. Both.
Yeah, I'm less sure about that second one too. Bacteria do have ways of communicating through like population density. They excrete compounds into the environment, and once they reach a certain what's the word? Concentration that can activate other things. We call this quorum sensing. So like once you reach a certain population density, bacteria can change their behavior and whatnot. I don't know that,
charge at the same time.
Yeah, exactly. I don't know that the fertility factor is doing much in the way of that kind of communication. But it is helping them spread resistant factors, these R factors. Okay, and it increases the chance of that spreading. And so if you've got the fertility factor, you could also pass that on So let's say that this is the F positive down here on the left. It grows this pilus over, conjugates with an F negative, and it sends a plasmid. Okay. On that plasmid could be the R factor of interest at that time, so resistance to some antibiotic, but it could also be the F factor itself, because this tube is just made out of proteins and such. And so if you send the code to make the tube over here, then this one's now going to be F positive, and it's going to be able to make its own conjugation tube. And so it can spread through a population really quickly. Okay, so it's pretty widespread because it's beneficial. It helps fight against negative selection by the environment. Okay. Now, I told you a few minutes ago that plants are photolithoautotrophs, and that it's just like cyanobacteria. And there's a really important reason for that, and it's this. Okay. So if you bust open a leaf, take out a cell, crack it open, what are you going to find in there, inside a leaf cell? Oh, you got a nucleus, you got a water vacuole, you got food storage. But how do leaves do photosynthesis?
Chloroplasts.
Chloroplasts. Okay. So you look inside a leaf cell and it's got chloroplasts. And if you zoom in on a chloroplast, you'll notice that it has its own chromosome and that it's a circular chromosome. What am I getting at here? Look at this tree.
Chloroplast is similar to a bacteria.
It is a bacteria. Quite literally. Okay, so we sequence that chromosome and build a tree from it, and it doesn't fall anywhere near plants. Plastids, the chloroplasts here, tree out with cyanobacteria. So inside of every chloroplast on Earth is a circular chromosome that looks very much like a cyanobacterial chromosome. And so here's nostoc, the one you looked at in lab. Really closely related to it are chloroplasts from things like wild mustard and rice. Okay, so all these plants you know and love are harboring cyanobacteria inside their cells. That's how they're doing photosynthesis. It's not the plant that's actually doing it. It's bacteria living inside the cells. That's kind of nuts. Of course, you look at, say, an animal or an amoeba or whatever, and you break open an animal cell. What do you find inside? You got your nucleus, you got some vacuoles, food storage, and what?
Mitochondria.
Mitochondria, the powerhouse of the cell. Everyone remembers that from like 5th grade and nothing else. It's the powerhouse of the cell. Great. That's a bacterium as well. Okay. So it trees right out with alpha proteobacteria. So your mitochondria has a different genome than your nucleus does. And it's a circular chromosome. Okay. So what we're talking about here is endosymbiosis. We'll get back to it in a second, but I want to put it in your mind here. And holy crap, we got like 5 seconds left of class. So let's just put a pin in this, finish up on Wednesday, starting with endosymbiosis. So any questions about our material so far? None?
Can you briefly go over again why it's beneficial for the, I think, fertility to be widespread? Or the...
Yeah. So what it comes down to is that if you've got the fertility factor sitting on a plasmid somewhere in your cell, you can transcribe that and then translate it into proteins. those proteins make this pilus, this conjugation tube. Okay. Now that's good because other members of your species in your population around you, you can send them useful stuff. Okay. Now, if you're sending them useful stuff, you're actually not picking which little plasmids go through the fertility factor. It's just kind of random chance what goes through. And so you might also be sending, along with resistant factors, the fertility factor. And so you can kind of infect the cells around you with the ability to also make this tube. And since it is so good at transmitting good stuff, it's selected for by natural selection.
Oh, okay. That makes more sense. Thanks.
Yeah. All right. Anyone else? You're free to go since we're basically out of time, but
Question about the pilus.
yeah.
So do the cells is that always just hanging off of the cell, or do they grow a new one each time?
It grows. Yeah, grows a new one.
How long does that take?
I don't know, but I do know how long the conjugation lasts on average is just a few minutes.
Oh,
and
okay.
so I don't know how long it takes to actually grow that and hook up, but then you got a pretty short window to transmit stuff. I mean, if you're dividing every 30 minutes, you're not going to stick around hooked to somebody else for too long. Okay, so you conjugate, DNA starts being passed actually back and forth along this thin little tube.
Is it possible for multiple cells to conjugate at the same time, like 3 or 4?
I haven't seen that. I wouldn't think it'd be difficult to find, actually. Yeah, the microscope that I typically use is not powerful enough to like see a pilus, and so I haven't personally seen it, but I don't see why not. It's not like they have, free will and are thinking about who to conjugate with. This is all just like enzymatic reactions that happen because of physics and chemistry. And so they're sending out tubes, they're growing towards a higher concentration of something that their fellow population member has. So I don't see why not.
And so is that an example of the chemotaxis?
Yeah.
Okay. So that's like how they communicate. They just move towards each other.
Yeah, and so different species are going to be doing different things and have different chemical signals and all that. And so it's really tough for me to generalize. We're talking about millions of different species that all have very different phenotypes, but in general, yes, I think it's safe to say that bacteria do a lot of chemotaxis. They do a lot of cell-to-cell signaling with just different molecules also. Some may make them move away from each other. Some may make them come closer together. Some may direct the pilus. I'm a eukaryotic biologist, so this is kind of a bit out of my wheelhouse. I understand the molecular side of things, but as far as like individual bacteria taxa go, I have pretty limited knowledge, except for big groups. Yeah. Okie dokes. We'll see you back here Wednesday. Have a good one.
Thank you.
Thanks.
Thank you.
Thanks.